In the last class, we talked about the laboratory trial of testing the seed stimulant effect of iron pyrite, where we use chickpea as a model system to exhibit the effect. So, these were the conclusion what we drew. Germination percentage goes higher, root growth is higher, shoot growth is higher, and overall biomass goes up, and sulfur percentage in those plants were higher, but iron remained constant. Now, from here, when we talk about overall biomass got higher, it means your carbon assimilation is higher. Now, from here, we talked about the how to translate this technology to the field. So, is it translatable to the field or to the farmers? What is the cost involved, and does this treatment has any undesirable effect? And in order to address this, we needed to have a field trial, mechanistic understanding at the ionic level, and long-term genetics study, which will of course continue, and the minimizing or optimizing synthesis cost of pyrite. So, the selection of crop which was made was spinach, and it is a short term crop and no fertilizer was used. So, the way it was done was, again you take the spinach seeds just the way chickpeas were taken. Again, these were divided into two categories: control and test, and those were treated overnight in water, DI water, deionized water, and DI water plus FES to suspension. Okay. Here you are having the FES to suspension. Here you are having the seeds. Now, after 12 hours of treatment, next morning, so you put the treatment in the night, next morning seeds were washed. Seeds were washed thoroughly, and now already your field is prepared. What you do on those plots, so you select 3 plots for control, or 3 or 4 plots for control, and 3 or 4 plots for test. Same condition, same soil testing values, everything same. So, they are getting, so one has to keep in mind there both of them have equal nutrient status, okay, then equal sunlight, equal water, and as we decided, no fertilizer, whatever organic matter is there. So, equal organic matter. Okay. So, everything was kept the same, distributed in a way. Now, what you do, you have those treated seeds sown at a distance, similarly. So, here you are having the FES2 or pyrite treated seeds, and here having the control seeds this side. Pyrite treated seeds exactly the same way all the repetition. Now the crop was allowed to stand there for like 60-70 days before the first harvest takes place, okay, which is essentially whenever you grow spinach, after 2 months you take the first cut. Sometimes people does it with 50 days, sometimes 40 days depending on, but it varies, but you keep it constant, okay. Then what was observed is, here are the results which emerged out of this study. The leaf size, see the leaf size of these ones, they are bigger than the control, significantly bigger leaf size. FES2 seed treated. Okay, so leaf size goes up. The first thing which was observed. It's a very interesting observation to see the leaf size has gone up. Second, the overall biomass went up. Next, one of the very interesting observation which was made was, in these leaves, chlorophyll percentage goes up. Now, when you talk about the chlorophyll percentage going up, that essentially means these plants which are seed treated with iron pyrite has higher ability to harvest sunlight. So, they are harvesting more sunlight. So, the third thing. Apart from it, manganese goes up. In sync with the previous result, sulfur goes up as compared to the control. Okay. Now, that showed that this is a long-term effect. So, the effect, if you remember in the last class I was telling, if I go back to the last class, is this effect a prolonged effect or is it just concentrated into germination? Say, apparently look like that this effect is a prolonged effect. This effect is not only germination. So, of course, before I get into that, so basically the germination part I did not talk about. So, the germination assay clearly said germination percentage is significantly higher on the FES2 treated as in sync with the chickpea results. So, leaf size went up, your biomass went up, your chlorophyll percentage went up, manganese went up, sulfur went up, your calcium went up, which was very interesting. So, nutritionally they are much more fortified with some of these compounds, manganese, sulfur, calcium. And as we have already discussed, when before performing the test, the nutrient status was kept same, sunlight was same, water was same, no fertilizer, and just grown in organic matter, but equal amount of organic matter. So, what is that which is changing the fate of these seed treatment? And the fate so much so that this effect is so prolonged that, like, I mean, 60 days the effect is being seen. And most stunning was this part. Chlorophyll percentage, how that goes up. It means there is something very unique which is happening. But first, let's try to answer how the germination percentage is probably going up. Will come slowly with each one of these effects. So, what really a seed is, if we look at it? A seed is a dormant life. Okay. This dormant life, when it gets water, it springs into life. In other words, it germinates and something like this. And we all take germinated seeds, those who all are health conscious, they do it every morning. Now, what water does? There are two questions here. One, how the seed maintains its dormant life, and how water changes that dormancy, because that seeds are kept in dark. You cannot keep seeds in moist places because they will go bad, or they will germinate and then they will go bad, or they will get the fungus attack. So, when we talk about a dormant life, what it is that makes that life, because it's a dry seed, if you see a seed, that needs a lot of imagination to wander, and the very moment you put it, it is going to give out it's as if it's a self-assembled machine. The machine will act whenever it will get its trigger, and the trigger is water. Now the question is, very fundamental question is, what keeps the seeds, what does not allow the seed to germinate? So it has been observed over the years that there are series of antioxidant enzymes present in the system of the seed, which antioxidant enzymes which creates a kind of a fort inside the seed, preventing it, antioxidant enzyme preventing or not allowing it to germinate. So, by the same logic, if the antioxidant enzymes, so antioxidant enzymes, as we know in the biological system, there are three major antioxidant enzymes. You have catalase, superoxide dismutase, or in short they call it SOD, peroxidase, so these antioxidant enzymes prevent the seeds from germination, but then by the same token, if you could deactivate them some way or other, you have a mechanism to deactivate them, then the seed should move towards germination. Where the logic, if something which is preventing the germination, if you could deactivate that switch, it will lead to germination. One of the studies back in 1976 exactly showed this result, that by deactivating the antioxidant enzyme, you enhance the germination. or you use a substrate. So how you deactivate them? You have to have something, like, you may use, say, catalase inhibitor, you use a superoxide dismutase inhibitor or a peroxidase inhibitor or something. Okay. So this is the first catch which triggered us to explore this pathway is iron pyrite aqueous suspension. I will take you back to that very first slide where I showed this. Told you that I will come back to this one. Does this milieu does something of that kind of stuff? Because as I told you, your major player is FeS2, fine, but then the challenge is with these ones, H2O2, which is an oxidant. Okay. You have Fe2 plus, Fe3 plus, very trace amounts though. Then you have SO4 minus, and then you have higher quantity of protons. Who is playing the role among all this? Now, most likelihood, the one where we put our guess was this one. Is the first one who is doing something interesting, because hydrogen peroxide is an antioxidant molecule, as well as it is in a trace amounts, it is a profound signaling molecule, and it is a very powerful metabolite. How to test what hydrogen peroxide is doing? So this was the juncture where the field trial is showing a great result, but this needs further explanation, that what that trace amount of H2O2 could do, how that possibly could breach, or how that possibly could break the antioxidant molecule bridge to get into the system and do something which we never thought of, and as a matter of fact no one thought of. An antioxidant nanoparticle suspension, which is not getting inside the seed, just by a surface reaction could do something way more profound than one can think of. Okay. So our journey will continue in this line, that how this trace amount of H2O2 and trace amounts of these molecules brings about a prolonged or a long-term effect in plants' life. Okay. Thank you.